Rickettsia Rickettsii by Daniela Russell and Kiana Barstad. Rickettsia are a diverse collection of obligate intracellular gram-negative bacterium that use ticks, lice, fleas, and mammals as its biological vector. In other words, the bacterium is carried within these arthropods and mammals. In 1906, Howard Taylor Ricketts found one of the more pathogenic strains of the Rickettsia genus in Bitterroot Valley, Montana, part of the Rocky Mountains. He found the bacterium Rickettsia rickettsii, which causes the infectious disease Rocky Mountain spotted fever. Rocky Mountain spotted fever starts with nonspecific symptoms of fever, headache, and vomiting, making it very difficult to diagnose at its early stages. 14 days later, red rashes appear first on the ankles and wrists, and then spread to the soles of the feet, arms, and torso. If left untreated, this disease can be life-threatening and cause neurological complications. Now that we've been introduced to R. rickettsii, let's go into some specifics. One of the R. rickettsioi's survival mechanism is preventing early apoptosis in the host cell. Apoptosis, or programmed cell death, is an important part of our immune system, used to kill human cells that have been infected by a foreign invader, such as a bacteria or a virus. By killing our own infected cells, this prevents further replication and spreading of the foreign invader. To understand this process, let's start at the beginning. An infected tick bites a human. The R. rickettsii bacterium enters the bloodstream and infects endothelial cells, smooth muscle cells, macrophages, and monocytes. R. rickettsii enters into these host cells by induced phagocytosis. Once into the host cell's cytoplasm, R. rickettsii phosphorylates and degrades I-kappa B alpha, which is an inhibitory protein. This activates the host cell's enzyme NF-kappa B. NF-kappa B sounds familiar, doesn't it? Go deep into your memories of genetics class and recall NF-kappa B is a nuclear factor or protein that controls transcription of DNA, influences apoptosis, cytokine production, and helps regulate the immune responses. As a transcription factor, NF-kappa B regulates the expression of genes through transcription. NF-kappa B makes sure the cell is expressing the right genes in the right cell at the right time. Once NF-kappa B is activated, it turns on the expression of several genes: FAS, BCL2, C-flip, caspase, IAPS, BFL1, and survivin. These are all anti-apoptosis factors that do exactly that, prevent the cell from undergoing apoptosis. Basically, the bacterium is turning the host cell's protective mechanisms against itself, almost like a double-ended sword. This allows R. rickettsii to maintain its cozy host cell environment while replicating and spreading. It's pretty crazy that a microscopic unicellular organism is capable of turning our own cell's mechanism against us.